Good morning, everyone. I'm Joyce, and today I'm going to talk about spatial temporal distribution of shrimp assemblages in the western coastal waters of Taiwan at the Tropic of Cancer, Western Pacific Ocean. And let's start with the introduction. First, let's talk about background and importance. Crustaceans are highly sensitive to temperature changes, affecting their distribution and migration. And second, Taiwan is located at the boundary of tropical and subtropical regions, influenced by Kuroshio Branch Current, South China Sea Surface Current, and China Coastal Current, resulting in significant seasonal temperature variations. And the picture on the left-hand side shows how the currents flow, and this one is South China Sea Surface Current, and this one is Kuroshio Current, and this one is Kuroshio Branch Current, and both of them are warm water, and only China Coastal Current is the cold one. Yeah, and on the right hand side is the picture on the right hand side shows 7 sampling sites, and we'll talk about it later more detail. Okay. Secondly, research objectives. In order to investigate the spatial temporal variation of shrimp assemblages in the western coastal waters of Taiwan and analyze key environmental factors affecting their distribution. And they examine whether global warming is driving shrimp species northward. And number 3, research hypothesis. First one is seasonal current changes influence shrimp assemblage, composition, and distribution. Second, tropical shrimp expand northward with warm currents, while subtropical shrimp move southward with cold currents. And next, let's talk about materials and methods. First, study sites and sampling period. First is May, and second is November. It's they're separated. They're late spring and late autumn in 2019, and covering 7 sampling sites. I can show these pictures here. It's from north to south. The first one is Wuqi, and second one is Wanggong, and third one is Taixi, and the fourth is Qigu. Next one is Jiading, and next Lingyuan. Last but not least is Fangliao or Fangliao. I don't know. Okay. And second, the methods. They used beam trawl sampling to conduct the experiment. They used 6-meter wide net and trawling for 30 minutes per site. And number 3, environmental data collection. Water temperature, salinity, sediment grain size, and nutrients such as phosphate and nitrate. Results and discussion. First, spatial temporal variation of shrimp assemblages. The first one, 27 shrimp species recorded, with 20 species found in autumn and 15 species in spring. And in the north area is cold water zone, and it's dominated by subtropical shrimp, like 哈氏仿对虾. It's pretty hard to pronounce in English, so I'll told you in Chinese. and in the south part, in a warm water zone, it's dominated by tropical shrimp, such as 波罗门赤虾. Okay, and let's talk about temperature and sediment affect shrimp distribution. First, tropical shrimp prefer higher temperatures, and it's more like definition of high temperature. It's higher than 25 degree and finer sediments. And on the other hand, subtropical shrimp prefer lower temperatures, and it's lower than 25 degree and coarser sediments. Number 3, relationship between shrimp assemblages and ocean currents. Warm currents such as Kuroshio Branch Current, South China Sea Surface Current promote tropical shrimp expansion. And cold water such as China Coastal Current favors subtropical shrimp dominance in the north. And this kind, 哈氏仿对虾, has shifted 0.5 degree northward over the past 20 years, indicating that climate change impact seriously. So here's the graph. The graph on the left-hand side is on the x-axis, it's sea temperatures, and y-axis is abundance. And here, these three, they represent for 3 different types of shrimp. Their names are here. And let me just explain one graph for you, and the another is the same concept. So let's use this MPAL for the example. It's they live in this area like 26 to 28 degree. Yeah, so it's more like tropical one. Yeah, and on the right hand side it's the same concept. So yeah, and these 2 pictures shows that in late autumn and late spring and late autumn, the period they sample. Yes, and you see this, the blue line is 25 degree of sea surface temperature, and on the right hand side is here. And the black box include the 7 sampling sites, yeah, on the west coast. Okay. And these 4 graphs shows the environment that shrimps live. Like let's look at the graph A. On the x-axis, it's silt and very fine sand. So it's the range of the sand is 3 3.9 to 125 μm. And let's use MPAL for example again. The range of it is from 40% to 80%. Yeah. So these four graphs all show that the environment the shrimps like and where they live. So graph A is silt and very fine sand, and graph B is fine sand content, and graph C is medium sand, and graph D is organic matter. So, and all of them on the y-axis are abundance. So, yeah. And let's talk about conclusion. Here are some key findings in this research. The first one is seasonal temperature variations and sediment characteristics significantly impact shrimp assemblages. And number two distinct shrimp assemblages are influenced by ocean current. Number three, global warming may drive tropical shrimp species northward, affecting ecosystems and fisheries resources. And last but not least, long-term monitoring of shrimp distribution shifts to assess climate change effects. Further analysis on the relationship between temperature, sediment, and shrimp reproduction. Thank you.
May I ask you why is finer sediment and coarser sediments? Can you explain it? Because I don't understand.
Finer sediments is much smaller, and the coarser one is much bigger.
Okay.
Yeah.
Could you pronunciation how to the pronunciation the hash foundation? I'm quite curious about it.
Parapeneopsis hardwicki. Is that correct? I don't know.
Can you explain the graph B?
Graph B?
Thank you.
Okay. Let's use MPAL for example again.
Okay.
It's fine sand content. It means that the small one, right? And most of the crust are focused on here. So they love the environment they live is like no fine sand content or less than 40%. Yeah. So
Thank you.
Just for this page, please do the conclusion of which type of string has the most abundance in fine sand or in medium sand.
let's look at MPAL again. MPAL in the graph A, it's silt and very fine sand. So, and it occupied like almost 80%. So MPAL, this kind of shrimp, loves the environment of very fine sand. Yeah. And number two, PHAR, and that kind of shrimps, maybe they live in like 50% and 50% in fine sand and medium sand. You know, you can say that it's like focus on here and here. So yeah. And the third one, PSIN. Yes.
Okay, thank you.
Okay, so first of all, next time please add page number.
Okay.
because it's easier for me to take notes where you can't make improvement. And then second, I think for the introduction part, so basically in this presentation, you didn't add any image outside the figure in the paper. So this makes this presentation not so interesting for the audience, because whenever I see a slide like this, unless you are a Nobel laureate, 除非你是诺贝尔奖得主, otherwise I won't bother looking at this slide. That's too much text, simply too much. Okay, for example, this is completely violating our guideline that you should have text less than 2 lines. This, you have 3 lines. And furthermore, so if I were you, I would, for example, where is that? For example, I don't know whether everyone recognize this term, crustacean. Okay, so you should be sensitive to any words that you think might be new to them, and then you should add image, because this one can be very easily explained. And then you have these slides where the map show the location and the type of currents mentioned in this slide. This is not a good practice. So if I were you, I would, this sentence in one slide, here, an image of crustaceans. And then this sentence, condense it, and then put it together with the map here. So you should have your text and your map side by side. And in this slide, if unnecessary, you should not put this. This is unnecessary. When you only talk about currents, you don't need this one. Because later you will talk about your study area, and what you did was you scroll backwards. This is also again back practice. You should always put the slide that or the image that you need next, immediately in the same slide or immediately after the slide, okay? So this one, put it in your material and method, okay? And of course, you should use mouse more often, not doing things like this. We don't know where you are pointing at. And then objective is okay, but because it's, again, this is long. In this case, please highlight keywords. Okay, use different color. Then, since you have hypothesis, a better way to always make better coherence of your talk is to come back to your hypothesis in your conclusion. So your conclusion slide should be your answer to your hypothesis, whether this one is correct and whether this one is correct. You answer them, okay? Not just conclusion. Material method. So here you have, the map that you show next to the current in the previous maybe 5 slides before, you should put it here, okay? Together with this, okay? This is funny, okay? And then, again, as I mentioned many times, the best way to explain all this, actually you don't even need this. What you need is a photo of this tool. That's all you need. And, okay, so, and then the major problem of this talk is you actually tell us the conclusion first before you show the data. So the flow of logic is totally in the wrong direction. What you should do is you should show data first, then you should say, from this data, what kind of conclusion we can make. Otherwise, this is a little bit like politics. You say things without any evidence. It's story, it's politics. It is not science, okay? So next time, please show data and then conclude each slides where you show data. Okay, what is the main observation you can get from this figure? Not all these conclusion and then you show data. That's not correct. And then here, there are a few tips. So here, I know many of you who present biological paper, which may have the same issue. What you should do is you only say subtropical shrimp, or you can even say A shrimp, B shrimp. You need to give them a very easy way to remember. Okay, you don't even bother to read this or find Chinese names. You just say A shrimp, B shrimp. Okay, tropical shrimp, subtropical shrimp. And when you do that, later in your slides like this, you should tell us which one is your A, which one is your B. And actually, after A and B, I don't even know what this P sim is. Okay, is it part of the conclusion here? I don't know. It doesn't seem to be any of them, right? So what is the purpose of having P sim here? The black dots, I don't know, okay? So you have to figure out why the author put this one into the graph. And then here you have this finer and coarser sediment, which Cindy asked. Here, in this slide, you should tell us, based on the author, you have 3 size fractions. You need to tell us which one, according to the author, is defined as fine, and which ones were defined as coarse. Okay, these are all related to sediment grain size. So I guess this one is fine, and these 2 are coarse. You should use graph to provide definition, not just, okay, fine is fine, coarse is coarse. You should use data to provide your definition here. Definition here, okay? Good, and again, print your conclusion next to your data, not before your data. And then actually, what was missing is, I don't really know why you present these 2 maps. I don't know. Okay, you need to figure it out. And there was no concluding remark. And you just introduced all this, what this 25 contour mean. You didn't tell us what is the purpose showing the map. Do it next time, not here. I don't know why you show this, okay? Good, so conclusion, go back to your hypothesis. Good, so I hope to see your improvement next time, so let's thank Joyce.